Alrighty, here we go. So last time we talked about cell cycle, right? So what are the three main phases of the cell cycle?
Interphase, meiosis, cytokinesis.
Yeah, so it's interphase, mitosis, and cytokinesis, okay? And what is the function of cell cycle?
To grow and repair.
Yeah, grow and repair is correct. That's the function. Now we're going to deal with meiosis. And we have a little bit of vocabulary to go over. So here is the study guide for that. So we're going to go over this, sort of, in detail, but you have an activity today that you'll do where you'll get the details for all this. I want to hit the high things. So at the very top of the study guide, it says sexual reproduction. We already defined what sexual reproduction is, right? Yeah, so what is sexual reproduction?
It's like sharing or like
Not sharing.
transferring genetic.
Good, transferring genetic information. Usually I just say the transfer of DNA. That's it. So the reason why we have that is because it increases variation. So if you look around the room that we got here, there isn't a one of us that looks the same. We all vary. Some of us have different color hair, different eye color, different skin tone, different heights, different muscle mass, those kind of things. That variation comes because of sexual reproduction. I want to skip down to the bottom where it says asexual reproduction. Okay? And now at the bottom, this is most unicellular organisms. So this is bacteria, prokaryotes. And it is some organisms that are eukaryotic. And so if you look on there, they have got three main parts for asexual. So asexual and it's not very complex, but asexual, you've got binary fission. I put too many S's in it, I do that a lot. Nope. Binary fission, this is when one cell basically splits into two. That's all it is. It's not really mitosis, okay? It's not really the cell cycle. It's just one cell splits into two. Are we okay? Everybody all right? You need an example?
No.
All right. That's one way of doing it. So that's one of the columns there. Budding. Budding is kind of fun. And this is like yeast. Yeast actually have more of a club shape. That's not a good yeast. They have a club shape look to them, like a club, like smack people with.
Oh. Okay.
So what happens is a little bitty one pops off. It would be like one of you suddenly starting growing maybe another little you, and then it just kind of pops off and it's its own little person and runs around. That's budding. That's yeast does that. Yeast does that. That's your best example of that. And then the last one, I'm introducing this term. The honors program needs me to introduce the term because we talk about it in a little more detail when you get to AP, and that's parthenogenesis. And here's the thing about this. It's a little weird. All I need you to understand is that it's asexual because there's no DNA that's being transferred, and that it basically is the female develops an egg into an organism without having any DNA being transferred to the female. That's what it is. So some lizards do this. Some sharks do this. Instead of having DNA transferred from the male to the female, the egg actually just develops into another organism. That's really all it is. And you just need to understand that definition. You don't need to go any deeper in that. That's when you get to AP, you'll get deeper in there. And it gives you samples of bees. Offspring are produced by females without any genetic contribution to males. It's on that study guide. That would be the best probably way to do it. I had a lizard that did this. You know, I had a bearded dragon. She did this. And she laid several eggs. And she hadn't been near a male for a long time. And when I asked the vet that I was working for about it, it said, well, there's a chance that she could have held on to some DNA from a male. But he said, chances are this is parthenogenesis for this particular organism. So anyway, at least that's what the vet told me. That's really the bottom part. Now, is there any DNA being exchanged between organisms with binary fission? No. What about budding? Is there any DNA being exchanged? No. All of these are asexual because no DNA is being exchanged. Clones. You know clones? That's asexual, right? You're just taking a cell and you're having it make another cell. This does not contribute to variation. Okay? This does not contribute to variation. So that's the big thing you need to take away there. So that's one major concept for today. You got that? I mean, it is literally just this straightforward. There's not any major detail behind this like some of the other things we've done. Good? Okay. All right. So the next thing then is the meiosis. And again, you have a activity today. I doubt you'll finish by the end of class, but you have an activity today that you guys get to do, and it will walk you through the entire details. I just want to hit the high points. And so to do that, I need to make sure that each of you has some fuzzy sticks. Yeah, grab some for. Did you get some? White, red, what? I don't care what color you do. All right, so here we go. I need some fuzzy sticks too. Here we go. So, meiosis has two parts. Meiosis 1 and meiosis 2. Meiosis is how you make gametes. Okay? And what are gametes? Egg and sperm. So those are the two gametes. So far, so good? Okay. Now, before I get into the details of this, here, actually, we'll do a little bit of details, but before I get into major details, and I'm not getting to major details, but before I get into this piece, I need to go over a couple of things. So first of all, in meiosis, very similar to cell cycle, what is the first stage of cell cycle where the cell rests and spends most of its time? Interphase. So some teachers, some professors say that there is no such thing, or meiosis does not start with interphase. It starts with, what's after interphase? Prophase. Mitosis, right? Prophase. Says it starts with that. What I'm going to teach you is that there is an interphase 1 because that's kind of what AP is looking at right now. They'll give you credit for either one from what I understand. And then there's prophase 1. Now, I want you to skip some space here. What comes after prophase in cell cycle? Metaphase. Yeah, we're not doing the prometaphase and all of those steps, so just leave those out. So then there's metaphase. Notice I left some space. What comes next? Anaphase. What comes next? And then what comes next? And it's telophase 1 and then cytokinesis. So this process divides the cell. And we'll talk about what that means in just a moment. In meiosis 2, there's prophase 2. Notice I didn't put interphase first. Okay? What happens in interphase? What happens in interphase, the S of interphase?
Like DNA.
DNA replication. So if I don't have an interphase here, do I replicate my DNA again? No, I do not. And then cytokinesis. All right, I'm trying to make this quick because the meat of this is in the activity that you will do. Okay? All righty. How many chromosomes do you have in your body cells? 46, right? 46. How many of those chromosomes are paired? All of them are. So there's 23 pairs. There's 23 pairs, okay? So one set of chromosomes came from who? Your mom. And one set of the chromosomes came from your dad. Good. So that means you have 46 total. So far, so good? And 23 came from mom, and 23 came from dad. Everybody all right with that? We okay with this? So that's how you got your 46. That's you. We good? Okay. So that means there are 23 pairs. When we talk about the number of sets, how many chromosomes are in the number of sets, we use the words haploid and diploid. Haploid is in number of sets, one set. So your haploid number is 23, because that's just one set of the chromosomes, right? One you got from dad or one you got from mom. Everybody good? Diploid is called 2N, and that means there's two sets of chromosomes. One set from dad, one set from mom. That's 46. Okay? Everybody okay? So if I said a rabbit had 23 total chromosomes, its diploid number is 23. What would its haploid number be? Or excuse me, 24. What would its haploid number be? It'd be 12. Does that make sense? So it takes two gametes, an egg and a sperm, to make an organism. It takes two haploids to make a diploid. Is everybody okay with that?
Yeah.
All right. Haploid is one set. So you have two sets of chromosomes. You got a set from mom and you got a set from dad. Diploid is both the sets together. So if I say the diploid number is 46, then your haploid number, the number of chromosomes that needs to be in either egg or sperm, has to be 23. Are we good? Okay. So that's second most important thing you need to make sure that you've got down. All right? And then this is the last thing I'm getting ready to do. Give you a minute to write things down that you need to.
What's issues?
I didn't realize I was out over there. So, okay, and again, these are the steps. You have to have the 1 after all of these because that tells you the first time the cell gets divided. And you have to have a 2 behind these because that tells you the second time. Cytokinesis, you don't need to put a 1 or a 2 behind. Okay? All righty. So, what happens in the S phase again? Yeah, DNA replication, you synthesize DNA. What's going to happen is we're going to synthesize that DNA, but our number of chromosomes don't change. So here's where these guys come into play. All right. Just for sakes of arguments. So let's say a cell only has 2 chromosomes. So far, so good? Okay. This is the paired set. So here we are. One of the chromosomes came from, one of them came from, okay? In the S phase, these chromosomes, they get copied. But I don't have 4 chromosomes. Okay? So the reason why I gave you the fuzzy sticks was so that you could kind of play along. You don't have to, but it's just what we call manipulative to help you be actively engaged in what's going on. All right, so how many chromosomes do I have? 2. These things stay, they're not called a chromosome until they split apart. So if I told you the diploid number was 2 for here, I'll erase that not to confuse you, then the haploid number has to be 1. So in prophase, what happens is the tetrads align. In other words, the homologous chromosomes, the ones that are the same. So for you all being 46, all your chromosome number 1s, your 2 chromosome number 1s come together. But because they have the chromatids on them, because they have the arms, okay, make sure you do the reading, they like to hang out together. Are we good so far? Yep. Diploid is 2 sets, right? Diploid is 2 sets, 2 ends.
Yeah, like, you mean, like this one? Or,
Diploid,
okay.
because there's only 2 chromosomes in here, but that's its diploid number.
Okay.
Okay? So then in interphase 1, during S phase, we make a copy of the DNA, but that copy isn't split. In other words, it's not that we have now 4 chromosomes. We still have 2 chromosomes, but now we have 4 chromatids, 4 arms. Everybody okay with that? And that's really important that you understand that, because these guys, so for you, all your chromosome number 1s come together, all your chromosome number 2s come together, right? They just pair up. And that forms a tetrad. Do you see the tetrad? 1, 2, 3, 4. 1, 2, 3, 4. We okay? Tetrad. Why is this important? Is because they're so chemically the same that sometimes the arms switch. Sometimes the arms switch places. And that switching is called crossing over or recombination. Now, why is that important? Because it causes variation. And I'll give you an example here. It's really hard to do this with the fuzzy sticks. It's really kind of hard to do with the fuzzy sticks. But if these are my 2 chromosomes, here's one of the chromosomes, and here's the other chromosome. Remember, this is the diploid number. If I had a gene that was gene A here and a gene that was B here, and the other allele on that same chromosome, maybe that was little a and this was little b, if these 2 arms switch places, this chromosome no longer is a big A and a big B on it, right? If these 2 arms switch places, if I made copies, so I'm making a copy here, so I made an exact copy of it, that's in synthesis. I made another exact copy of this one, I'll just make it red as well. So there's that exact copy, I just don't have room between. But if these 2 guys switch places, right? If this one now becomes this side, I have a little a here, and I have a big A here. Now these aren't exactly the copy, and I've mixed up the alleles. I've mixed up the type. Do we make sense? Okay. So, what happens in metaphase? What happens in anaphase? They start to come apart. Now, I have, let's keep going. And then in telophase, they come apart, right? Go to the poles. And then cytokinesis, they're two separate cells. So now I have two separate cells. I can't draw. I've got two cells. Oh, that was kind of cool. I got two separate cells. Are these cells haploid or diploid? Because they only have one. Remember, they're not considered separate here until they split apart. Okay? So if this was a human, you started off with 46. By the end, you should have two cells that are 23. This is called the reduction phase of meiosis, meiosis 1. Reduction, this whole thing. Right, the whole thing, because what we're doing is we made our copy, but that copy is still just the 46 chromosomes. When we get down here, those 46 then split apart just like we did here. Remember, there's still two, we're still diploid. When they split apart and become two cells, now it's haploid. There's 1 and 1, but those ones have two chromatids with them instead of just one chromatid. Okay, then we come over here to this side. So far, so good. Now, we don't have copy that we make. We go through the same process, and so now we're splitting the copies apart. So in the end, we're going to get how many? How many chromosomes? We're going to get 4. Right. Right, because it was like that. But we called that one chromosome. When we split it apart, now that becomes its own chromosome. This is its own chromosome, that's its own, and we get 4 different cells.
Which process is that?
Meiosis 2. So for people, we have 2 cells that are both 23, and what happens is we get to the end, and now we have 4 cells that are 23. And all 4 will not look like the original chromosomes because we had crossing over. Does that make sense? Yes, sir.
So meiosis 1 was also the 2 chromosomes, and meiosis 2 is also the 4.
So in meiosis 1 here, you get from a diploid number, whatever that is, to a haploid number. Okay? And then in meiosis 2, we divide up the homologous chromosomes. The homologous chromosomes, there's the ones that got copied, remember? So that's when we divide those up. So we now have a chromosome, but with just one chromatid on it. Does that make sense? And if this was a human male or a mammal, every one of these would become a sperm cell. If this was a mammal female, then only one of these would become the egg and the others would be really, really small. And then this is what gets released every time the menstrual cycle goes through. Got it? Details are in what you guys have your activity to do today. That is it. Mm-hmm. And that's it for me.